Heterodimers represent a recent research avenue within the wider field of opioid receptor signaling. Traditionally, opioid receptors have been thought to function as isolated units. In other words, the mu opioid receptor has been thought to mediate its effects by itself without interaction with any other receptors at the surface. In the last 2 decades, this view has changed, and it's now becoming apparent that opioid receptor interaction, so the interaction of one opioid receptor with another, may be important functionally. A heterodimer is the name given to describe a protein complex consisting of two different types of receptor. In this graphic, I have used the mu delta heterodimer as an example. You can see the individual mu opioid receptor in red in association with the blue delta opioid receptor. Whilst these still exist as distinct entities within the complex, they function as one. In other words, the functional, signaling, and pharmacological properties of this mu delta heterodimer are unique and differ from the properties of its parts. So the mu opioid receptor acting in isolation will have a different profile to the mu opioid receptor when it's in complex with the delta opioid receptor, and the same goes with the delta receptor. The relevance of this to our purpose of raising the hedonic set point has been shown by researchers identifying the distribution of mu delta heterodimers in vivo. To elaborate, these complexes have been found to be abundant in the nucleus accumbens and the ventral tegmental area, both hedonic hotspots, both important to regulation of mood, regulation of euphoria, as well as reward. Interestingly, or perhaps frustratingly, it has been unclear as to what mechanisms at the mu delta heterodimer lead to the various physiological effects that have been seen. These effects include reduced withdrawal and dependence, reduced tolerance, as well as hedonia, antidepressant-like and anxiolytic-like effects, all within preclinical rodent models. Whilst these effects have been seen across a variety of experiments, the agents used to induce them have varied and appear contradictory. For example, the delta agonist with a mu delta heterodimer selectivity, CYM 51010, was shown to reduce tolerance in the mouse model. In contradiction with this, the mu delta antagonist, D24M, was also shown to reduce tolerance, withdrawal, and dependence. How can we square these apparently opposing effects? An important issue here to consider is selectivity. D24M is the most selective antagonist that has been produced for the mu delta heterodimer. Therefore, we can be fairly certain that when we apply D24M to our mouse models, Any effects that are observed are due to the blocking of this heterodimer. CYM 51010, on the other hand, has delta receptor affinity as well as some mu receptor affinity. What this means is that we cannot be sure that it is the interaction of CYM with this heterodimer. We cannot be sure that it's the activation of this heterodimer that causes the reduced tolerance, rather than, for example, CYM interacting with the delta receptor. This reflects a general limitation within heterodimer research in the selectivity of the ligands and antibodies produced to identify mu delta receptor function. With more selective tools, we can more confidently say that blocking or activating the mu delta heterodimer will be the desirable option to produce anti-withdrawal, anti-dependent effects, as well as, in places like the nucleus accumbens, hedonia, antidepressant, and anxiolytic effects. More research is needed to elucidate these specific interactions at the mu delta receptor, ideally removing the potentially confounding interactions with the mu delta receptors them individually.